Hello everyone, my name is Jean-Christophe Lachance. I am a fourth-year PhD student from the University of Sherbrooke. Welcome to my poster presentation titled Genome-scale Modeling of Mesoplasma florum reveals key features of a minimal gene set. I will start by asking a question. How interesting would it be to make accurate predictions on cellular behavior 100% of times? I guess everyone who has been working in biology for a little bit would totally like that. To reach such a level of precision, though, we would need to understand every molecular functions within the cell. This is a context that drives our interest for minimal cell research, since minimal cells, with their lower number of components, are closer to complete understanding. We want to initiate systems biology efforts in Mesoplasma florum because it is a fast-growing and naturally occurring near-minimal cell. In fact, it contains 680 genes and has a doubling time of 40 minutes in rich medium. The model that we build here for mFlorum is standard flux balance analysis metabolic model. We started by identifying metabolic candidates using a combination of three computational approaches. First, we compared the proteome of mFlorum with that of 4 other molecules. Those are bacteria of the same phylogenetic group, for which metabolic models have been reconstructed, hence allowing us to get reactions that are conserved in closely related species. Second, we used the iTasker suite to generate 3D protein structures for 386 mFlorum proteins. This allowed us to make function predictions, not only from sequence, but also from structure. Finally, since they are very useful in metabolic reconstruction, we used the DTEC V2 software to generate probabilistic identification of enzyme commission numbers. We gathered all the information from these three methods in the Sankey diagram to provide the general level of confidence that can be attributed to proteins. Next, we reconstructed the entire metabolic network of mFlorum, which you can see in full right here. This network is composed of 208 genes and roughly 350 reactions. Key features that we notice are the general lack of anabolism in the cell, a complete dependence on glycolysis for energy production, and a substantial amount of import from the growth medium. In fact, the complexity of mFlorum does not reside in its genome, but rather in its growth medium. The original growth medium for mFlorum was the ATCC 1161 medium. This rich medium is composed of undefined components, such as serum and yeast extract, and was in fact so rich that it would allow growth even when no sucrose was added. This was problematic since we could not change the medium to test different phenotypes. So we set to simplify it and found that by adding CMRL 1066, the concentration of undefined components could be lowered to levels where no growth was seen when sucrose was added. We found 10 components specific to CMRL 1066 that are probably essential for growth. Then we defined the biomass objective function for the model using the macromolecular wave fractions and multiple omic data sets obtained in a previous study from our laboratory. These were used as input for the above software. Having the CSY medium allowed testing the assimilation of 14 different carbohydrates by mFlorum. Surprisingly, we found that maltose and mannose could be assimilated by mFlorum while it was not predicted by the model. This provided a basis to reevaluate the alternate carbon metabolism. Using the 3D generated structures, we were able to find matches with enzymes of known promiscuity that could explain the observed discrepancies. Next, we compared the model flux states and single gene essentiality prediction to experimental data. We reached a 78% accuracy for flux state versus gene expression and 76% for essentiality versus TN-seq data. We used the context provided by the false negative predictions to reevaluate some of the model constraints. Finally, we used a min genome algorithm from the Marinus lab to generate a minimal gene set prediction. This algorithm uses as input the transcription architecture, experimental gene essentiality, and the metabolic model, and iteratively finds the largest deletion that satisfies all constraints. We found that after 75 deletions, no more genes could be deleted, hereby reaching the minimal gene set at 552 kb and 519 protein coding genes. We compared the minimal gene set prediction to the synthetic minimal cell JCB ISIN3 and found that 57 genes targeted by deletions were homologous to the experimental cell. This suggests that multiple minimal genome scenarios are possible. We then sought to understand which cellular functions were most targeted by deletions. We found that proteins of unknown functions were targeted first, which fits our objective to reach complete understanding of molecular functions, meaning that by reducing the genome, we would mainly remove proteins that are not well characterized. In conclusion, we generated a metabolic model which revealed a metabolism with low energy requirements and highly dependent on import from the growth medium. Together with experimental validations, the model provided a context to re-annotate certain of the M4 genes. And finally, we formulated a minimal gene set prediction that we compared with the synthetic minimal cell JCB ISN3 and found potential discrepancies that would reveal an alternate minimal gene set. Our current efforts could be complemented by adding the gene expression machinery in the M4 model and formulating other, potentially more detailed predictions of minimal gene sets. I would like to thank the labs that supported this research, namely that of Professor Sébastien Rodrigue and Pierre-Étienne Jacques in Sherbrooke, as well as the Systems Biology Research Group led by Professor Bernard Paulson at the University of California in San Diego. Thank you to everyone who attended.